Do you remember the commercial, "Can you hear me now?" If you're having trouble hearing me right now, you may be part of the 25% of those over age 12 who have hearing loss in one or both ears. Hearing loss in humans is progressive and degenerative. It gets worse and worse over time. In the population over 70 years old, 50 to 75% have some degree of hearing loss. There is a type of cell in your ear called a hair cell. Hair cells are what detect sound as well as movement information like you're tipping. Unfortunately, these hearing and motion cells, the hair cells, can be damaged by many things, including loud noises, infections, and even certain antibiotics intended to cure those infections. But what if we could figure out how to regrow these cells? Maybe we could reverse hearing loss caused by cell death. In order to figure out how to potentially regrow cells, we first need to understand how parts of the ear become specialized. Imagine you have a blank canvas and paints. The canvas could become any painting in the world depending on where you put the paint. So directions might be helpful. Initially, the developing ear is much like a blank canvas, and I have been studying two directing signals. Part of their job is to affect what parts of the ear become hearing cells and what parts become neurons that will connect the ear to the brain. I have been studying how 3 genes respond to these directions, because changes to the directions, and therefore these genes and the proteins they make, may impact how much of the ear becomes painted with hearing cells versus neurons. So far, I have determined that one of the genes probably primarily deals with one set of directions, another gene deals almost exclusively with a different set of directions, and the third gene responds to both sets of directions and might help coordinate the two. This third gene creates a protein that is essential for hair cells to survive, but we don't know exactly why. Using molecular tools, I have constructed a genetic modification that will allow me to mutate the protein at very specific times. This will help me determine why the protein is so essential. These results help us better understand how the ear forms and could potentially point towards ways to regrow these cells. Hearing cells are unfortunately pretty easy to kill, and in humans they do not regrow, resulting in hearing loss that can impact an individual's social, cognitive, and physical well-being. But with my research, hopefully in the future, we can figure out how to regrow these cells and restore hearing so that you never have to yell at your grandmother again. Thank you.